I'm the head of customer service at Omicsoft, and so I think I've written to many people in this room. And so before we get started, I want to also point out that we still have slots open for the one-on-one sessions for both today and tomorrow. So if you have any detailed questions that you want to ask one of the customer support team, we can help you sign up for those. So I need to start by saying that these QIAGEN Bioinformatics products are for research purposes only. And today, the goal is to develop a better understanding of innate anti-PD-1 resistance. In particular, some of the goals that we're going to try to show you are identifying genes and differentially expressed splice variants that could be a good marker for anti-PD-1 treatment resistance, identify an actual signaling pathway that's revealed as a key to innate resistance, identify several biological processes that could be involved in this resistance, put these all together to identify a full transcriptional program that is detected specifically in non-responders to anti-PD-1, and finally generate an actual testable hypothesis about the kind of underlying biological mechanism and a way to actually revert this experimentally. If we're looking at the QIAGEN sample to insight workflow, we'll be focusing on the last two steps, that is data analysis, which is Omicsoft's strong suit, and then the higher level interpretation, which is the focus of IPA. As far as the actual steps that we're going to walk through today, I will be taking you through the basic RNA-Seq pipeline. As many people are aware, the RNA-Seq pipeline can start from managing just the raw sample metadata, raw and aligned QC and alignment, quantification and visualization of expression, and then identifying differentially expressed genes and transcripts. Because many people are aware of this pipeline and have watched the videos or read the tutorials, I'm not going to just kind of walk through all of the steps, but instead focus on a couple of the key features and maybe unknown or new features that you may not be aware of, some cool tips and tricks. And then the final step will be to seamlessly upload the data to Ingenuity pathway analysis, and then my colleague Jean-Noel Billaud will take you through the higher level analysis in IPA, helping with the biological interpretation of the transcriptome results, such as looking for expression signatures to identify significant pathways, look for differentially expressed splice variants, and then generate full novel regulatory networks to support a new hypothesis that could be tested in the lab. The overall goal in the initial integration between Omicsoft and QIAGEN has been integrating ArrayStudio to Ingenuity pathway analysis. What this means basically is that even if you have your own expression data, your own RNA-Seq or microarray data that you've analyzed within ArrayStudio, or if you want to take land data that have been pre-processed and are available in disease land or oncoland, and you want to explore these data further in IPA, it's only a click or two away, basically. As a very, very brief background on anti-PD-1, in essence, PD-1 receptor is expressed on T cells, and many normal tissues express PD-L1, the ligand, and this helps downregulate autoimmune response, and so you avoid autoimmune disease. Unfortunately, this also means that if a tumor is expressing PD-L1, it inhibits the immune system's ability to fight the tumor. And so the goal of anti-PD-1 treatment, or anti-PD-1, anti-PD-L1, is to actually stop this signaling, to have an anti-PD-1 or anti-PD-L1 blockade. So if we can prevent this immune inhibitory signal coming from tumor to T cells, then you could actually enable the immune system to attack the tumor. There are certain people who respond very well to this treatment, and there are others who do not respond very well. And it would be great to be able to actually predict the people who would respond versus not respond ahead of time, and that's what we'll be focusing on today. Oops, excuse me. In particular, we're going to be using data from this recent Cell paper, where they were analyzing RNA-Seq data, transcriptome data from 28 patients with skin melanomas. This was pre-treatment melanomas that were full transcriptome RNA-Seq, but then after taking the transcriptome data, they were subsequently treated with anti-PD-1, and just about exactly half responded and half didn't respond. And so it's a nice kind of hopefully predictive study where we could identify some inherent transcriptional signatures that would be able to tell the difference between responders and non-responders to anti-PD-1 treatment. Here's the basic experimental setup. 28 samples RNA-Seq paired end data, divided evenly between non-responders and responders. As many of you are already aware, there are always additional metadata that you can include. So in this case, there was also previous MAP kinase inhibitor treatment in about half of the patients each way. But we'll be focusing just on the non-responders versus responders in this morning's session.
So at this point, I want to get started with the actual demonstration with ArrayStudio, starting with a brand new feature where you can generate custom workflows. Many of you have seen in the ArrayStudio interface the Workflows tab, but most users would just click away because the workflows were predefined and very restrictive. We've added a new feature where you can define your own full workflow. You can generate a new workflow, group modules together, and add individual modules from any of the menus to your workflow. So if you hate diving through 3 or 4 menu levels to find a module, you can bring them to the front in your own custom workflow. So here's the ArrayStudio interface. Everyone is aware of the basics of starting a new project. So I've already generated a project, and I've downloaded the raw data from the SRA tool. I loaded the FASTQ files from the SRA tool into the RNA-Seq pipeline GUI. In the pipeline GUI, we can specify the genome, which was human B38, and gene model, ensemble R86. Chose parameters for alignment, which functions to run within the pipeline, and an output name and folder. And I've pre-run this analysis because it would take too long to run it live. We can see over here the output of the pipeline function. We have NGS data, which are pointers to the actual BAM files. We have omic data, which are count and normalized FPKM data at the gene and transcript level. We have a number of tables output, including raw and aligned QC metrics. You can also have exon junction reports, mutation data, fusion data, etc. And I've also pre-run a couple of statistical inferences that we'll come to later. Finally, I've pre-imported a couple of lists of genes because I'll be using these later in the demonstration. So after running the RNA-Seq pipeline, the first thing that you'll want to do is import your design metadata so that you can have more relevant information when analyzing your data. After running the pipeline, here you can see the NGS data design table, which just has the sample IDs, path to the BAM files, and alignment statistics. The traditional way to import metadata is to right-click on the design table and then choose Import. Then we'll find our tab delimited file. We'll append to the existing design table. And now we have all of the metadata attached so we can tell what each sample is. We could do this for each of the omic data objects as well, but I wanted to show you another trick of how to propagate all of these columns to any other table that has similar column names. This is called the propagate columns function, and we'll choose our source data, which is the NGS data, where we just attach these metadata columns. Choose the columns that we want to propagate, and then choose the tables that we want these columns to be propagated to. In this case, I want to choose all of my design tables for the omic data. Now we can see in all of our tables, we have the full set of metadata. So this can save a lot of time. So one reason why we output both count and FPKM data in the RNA-Seq pipeline is that most statistical analyses, like DE-Seq and Boom, are designed to be run on raw, unnormalized count data. But if you want to do visualizations on your expression data, you will want to use FPKM data, and in fact, you will usually want to use further normalization on these data and then perform a log transformation. So that's going to be the next step that we're going to take. Using my custom workflow, I'm going to select Normalize RNA-Seq data. And I'm going to choose my FPKM gene level data. I'm going to use another new feature based on a customer recommendation. So instead of having to find my data under a drop-down box, we can also use this tree to explore the data a little bit more cleanly. So here I want my gene level FPKM data and choose a normalization. In this case, I'm going to use upper quartile normalization because it's a nice simple normalization, basically targeting the gene that is expressed at the 75th percentile will be set to a scale target of 10, and all other genes for that sample will be scaled accordingly. So we'll specify an output name. And then the next step after we've normalized these data is to actually log2 transform these data. So we'll choose transform, make sure we're choosing the right data, and then we'll choose among the different transformation methods. In this case, I'm going to use log2 and a small pseudo count. Now that we've normalized and transformed the data, we can visualize these data. We'll just right-click on the omic data, choose Add View. Up to 40 different visualizations can be available depending on the data type. The real workhorse visualization is the variable view. This will plot a single row, a gene, or a transcript for every column, which is usually every sample, showing the expression level of the given gene. All views are extensively customizable, so I'll perform a couple of different manipulations on this data set to make it a little bit more visually appealing and also more informative. To start, we're going to go in the Task tab and choose Specify Title Columns to change from Gene ID to Gene Name, which will be a little bit more informative. Now we can see which gene we're looking at. Now we're going to reorganize each of the samples to group them by some of our metadata. In this case, I'm going to use our PD1 response column. Now we've grouped the samples based on whether they were non-responders to anti-PD1 treatment or responders. And of course, because it's important to label our axes, I'm going to label our y-axis here under Change Chart Properties, y-axis. In a minute, we'll manipulate some of the details of the plot with change fill and change simple. But first, I wanted to change from a simple scatter plot to a box plot. So under Change Profile Gallery, I will choose our box plot to give a nice box plot. Now let's do some manipulations on this box plot. First, we're going to change the fill on the box plot, choose Change Fill Properties, and let's change to maybe color the different boxes based on category, such as PD1 response. And we'll reduce the opacity a little so that we can actually see the data points. And now let's change the symbols to make these look a little prettier. Choose Change Symbol Properties. And we'll start by coloring them. Maybe color by another metadata column, such as MAP kinase treatment. And we'll change the shape from squares, which I'm not a big fan of, to circles. Maybe reduce the opacity just a little bit. And maybe we can add a little jitter here. So now we have a nice plot, and we can look for a gene of interest. So we can either scroll through the 58,000 plots that we have here for genes to find our gene of interest, or we can search for a specific gene, or we can use the extensive filtering capabilities to find the exact gene we're interested in. For example, we can look for E-cadherin or CDH1. And we can see immediately that CDH1 is much more highly expressed in the responders to PD-1 treatment compared to the non-responders. And since E-cadherin is important for cell-cell epithelial interactions, if you have an epithelial to mesenchymal transition in a cancer, you might see a downregulation of CDH1, and this is generally bad news because this is the kind of thing you will see in metastasis. So let's say we want to look at another gene, such as FGF2, so we can change the filter to find FGF2. We can see that with our current grouping, it's not very interesting, not a big difference between the non-responders and responders, but we can use our metadata to regroup these data. For example, it's been observed that FGF2 is differentially expressed in patients with mutant BRAF versus wild type, so we can change the grouping to observe this. The metadata included status for whether or not they were mutated for BRAF. And we can see that there are a couple of BRAF mutations that have higher FGF2 expression compared to wild type for BRAF. So I want to clean up this plot a little. First of all, there are a couple of less interesting BRAF mutations that I'm just going to hide for the moment. Use the filter column to hide certain observations. And now I'm going to collapse these three different BRAF V600 mutations into a single column to make it a bit of a cleaner plot. I'm going to select all of the V600 mutant samples, and then under selection, I'm going to choose Generate Column from Selection. Change the label to BRAF V600. Then indicate the selected are mutant and unselected are wild type. And we can change the plot again to profile based on our brand new column. So here we've just generated a new metadata column on the fly to make the view more clearly display the results, that there's a significant difference between FGF2 expression in mutant for BRAF V600 versus wild type. So let's move on to differential expression. We have two main modules for analyzing NGS data, RNA-Seq data for differential expression, and that's DE-Seq and VOOM. So regardless whether you're using DE-Seq or VOOM or even the microarray GLM model, the basic interface will look basically the same. And as we've mentioned before, we'll want to use the raw count data at the gene or transcript level. So for Jean-Noel's portion of the talk, he will be using the transcript level differential expression analysis, but here I'm going to use the gene level analysis, but you can use either depending on what your goals are. We will set up a simple one-way ANOVA comparing non-responders to responders. So here I'm going to open the DE-Seq GLM window, make sure I choose my raw gene level count data. First, set up my model, use PD-1 response, then I will specify the test. And basically, I want to compare to the responders as the baseline. So I will add my test comparing non-responders to responders. Simple one-way ANOVA, set up the analysis, and then either send to the queue, or if I wanted to, I could run this analysis locally. Since I've already run this analysis, we can go ahead and open this up. The output will include a table in the Inference folder. The Inference report will have log2 transformed fold change, linear fold change, as well as raw and adjusted p-values, and then finally, a group max column. What this is finding is between the mean values of your two sets of samples, your case and control, whichever value is greater will be reported here. And this is an important column for when sending your data to IPA. You will also find a Volcano plot so you can visualize the data. So let's perform a couple of manipulations on this Volcano plot to make it look a little prettier. First, we'll change it to quality first mode, and then choose change symbol properties. And I want to emphasize upregulated genes versus downregulated genes. So maybe I'll use the log2 transformed fold change, set the size to be by the adjusted p-value, make them a little bit bigger so we can actually see these data, and then change again to circles because I do not like squares. And then change it so that when I click on one or more of these dots, I can actually tell which gene is up or down regulated, and then reduce the opacity just a tad. So now from this plot, we can immediately explore the data, select a couple of genes, and see that there are some upregulated genes like FOXC2 and ALDH1L2. There are also some downregulated genes. So now that we've done this test, you might want to go back to your variable view and check out one of these genes to see what the expression pattern looks like, or you might want to select a group of genes and create a gene set with the list function. So to do this, we might want to select your genes, then right-click on List, and choose Add List from Selected Rows. Call the list name Some Interesting Genes. And now we've recollected our list of interesting genes here that we can use to then filter any of our plots or variable view or any table to explore these genes in more detail. So at this point, I want to move forward with the statistical analysis. Because DE-Seq2 and VUM have different statistical assumptions, it's generally a good idea to run both of these modules and then compare the results, maybe focus only on the genes that were found in both sets of significant genes. So one way to do this is to summarize the data using summarized inference reports. So we'll start with the DE-Seq report and do summarize inference report. We'll find our gene level DE-Seq data and then specify a cutoff, let's say only interested in genes that were upregulated at a log2 full change of 1 or downregulated by -1, raw p-value less than 0.05. Add this test, DE-Seq 0.05. But let's say we wanted to do a slightly more stringent test, so we can change this to 0.01, change some other parameters. Add this, DE-Seq 0.01, and then we'll want to output a set of lists based on each of the conditions. Then we can submit this analysis, and we'll get a couple of objects out. First, we'll get a couple of list objects that contain our significant genes at each of our cutoffs, and then we also have a list table. And this is an interactive table where you can click on any of these cells and get the inference report specifically for the genes that were passing these criteria. So we have our test of 0.05 and 0.01. So let's set up this same test for VUM. Summarize inference report, open up our VUM results, set up a condition, maybe set up a second condition, more stringent, output our lists. And now we can either use these lists directly, or we can actually compare the lists to compare between DE-Seq and Boom, or use our different cutoffs. So there are a couple of different ways that we can compare these lists. One way would be to generate a Venn diagram view. We can attach it to any of these visualizations. Let's use our UQ log2 transform data, add a new view, create a Venn diagram view, and then we will add our lists of interest. So in this case, if we wanted to compare our significant genes at the significance level of 0.05 for DE-Seq and Boom, just select your lists and drag them and now we have an indication of the genes that were uniquely differentially expressed according to the Boom test, DE-Seq2, and our intersection between the two tests. We can also calculate this intersection by just right-clicking on List, choose Manage Lists, then find our tests of interest, and then we can make a new list from the intersection between these. And now we have a new list that indicates over 600 genes that are promising candidates to look a little bit more detail. Now, before I submit these inference report data to IPA for further analysis, there was one more powerful technique that I wanted to show. It's a recent feature that we've just added, and it allows us to custom annotate any of the tables based on the contents of lists. So let's go back to our Volcano plot of the results of DE-Seq2, and let's say we had a list of genes in various gene sets. For example, gene signatures from another study. For example, I've got a collection of 3 different signatures showing different gene responses according to MAP kinase inhibitor treatments. And these are somewhat overlapping, but there are also unique members within each of these. And I want to see where each of these genes are within this result set. To do this, we need to annotate the underlying inference table for each of the genes, whether or not they are members in one or more of these. To do this, all we have to do is select one or more lists, right-click, and choose Add List Membership to Table. Find our target table, which is our DE-Seq result. And this is going to add a new column to the end called List Membership. If we open the full test and sort, we can see each gene that is a member of one or more of these lists. So one simple use for this is to just recolor this plot based on the membership in one of these lists. Change to categorical, and then find our list membership. And now we can see where each of the interesting list membership genes are in this plot. Can click on one, such as angiogenesis, and we can see which genes are upregulated or downregulated within this set of genes. We can also hide all of the missing genes, so we can really just focus on the genes within our list. And now we can see that the vast majority of the MAP kinase inhibitor responsive genes are upregulated in the PD-1 non-responders compared to the responders. There's a very strong trend of upregulation. This trick of annotating tables based on list is also really useful for annotating genes in, say, heat maps or hierarchical clustering to label clusters of genes using our color bar function and so on. It's really powerful, and I've been using it all the time recently. So the final thing I want to demonstrate is how to upload your data really quickly up to IPA. Once we have an inference report, all we have to click is Integration, IPA, upload data. We'll choose our data set that we want to send to IPA, choose the data type. So in this case, we want to specify gene symbol, maybe name our project name and data set. Select the specific test that we want to upload. We can upload up to 20 at a time. You can optionally send a full core analysis as well, so we can pre-specify the parameters for performing the basic analysis, and this can run in the background before you even open up IPA. So for example, in this case, we might want to specify a fold change of 2, p-value of 0.05, and a group max intensity of at least 10. So this is going to make sure that either the case or the control samples are pretty highly expressed to be a fairly restrictive test. Then we'll submit, and this will connect to IPA server and upload it to your login.
So I guess while Jean-Noel gets set up very briefly, do we have any questions on this, on the first half? No, we're good. Yes, PJ.
So I noticed you did something that many people do. You first go to quality mode instead of speed mode, and then you get rid of the squares and make it circles, and then you size by p-value. Is there a way to make that a default or a simple click? Because that ends up being, at least for me, those exact same 3 clicks every single graph.
So I think, so PJ was asking just for the basic manipulations, is there a way to set those as defaults? For the quality first mode, probably not currently, but the quality first mode is probably becoming less relevant as computers become more powerful. For the others, yeah, I think if you go into preferences, you should be able to set the defaults for different chart types. And if it's, I know that it's there for even the microarray options that we have in the table options, you can actually pre-specify the defaults for all of those different parameters. And if it isn't there, then we could definitely add it. Yeah, so I think it's a good idea if you, to just be able to set those in preferences. And it's there. It's another one of the things where you just have to find it in the menu.
Thanks.
Yeah. Yes.
ArrayStudio and IPA are separate.
Yes. Yeah, you have to subscribe separately. But I guess you'd talk to the QIAGEN salespeople about the details on that.
So I'm Jean-Noel. I came from formerly Ingenuity system for the IPA. So to answer your question right away, the data is sent. If it will be sent automatically, and you just have to log in if it was not logged in, and that's it. So for the people that do not know IPA, it's been a long story 15 years ago, and I'm very glad that we have met Omicsoft, and now we are really stronger together. So IPA, the IPA is based on a knowledge base, right? Knowledge base has acquired, is constituted by finding that has been acquired, for the last 15 years. And the findings come from papers, peer-reviewed papers. They come also from third-party database. And everything is adjusted, structured, and according to a certain ontology, we can have a very strong way of finding any type of scientific-based findings. And the findings are always decorated that way. We have everything that we know about for that particular genes. If it was a biomarker, what is downstream gene of that guy? Does it belong to any particular disease? Is there any function it belongs to? Any canonical pathways, any drugs? So we have that for every findings in our knowledge base, and we have around 5 million findings. And the good news is all the findings that we have are structured this way. They are very contextual, and we can understand the direction effect. Because of that, we can infer a lot of things. We can infer the increase, we can infer the decrease of a particular phenomenon, downstream or upstream. And based on that, this is the power of the prediction we have. So the question we have in IPA are multiple. We can answer which type of signaling pathways or metabolic pathways are they activated? Are they inhibited? We can look at what we call the underlying transcriptional program using the upstream analysis. We can look at what are the biological processes that are involved and in what way, because of that predictive power. We can say if apoptosis is increased or decreased, for instance. We can connect the molecule together, which is in black will be presented. In gray, I will not mention it today, just to show you what we can do. We can explore also genes or pathways in a network and other simulation using the molecular activity predictor. We can look at splicing variant of interest, as Joe mentioned, splicing variants, and we can do that using isoprofiler and isoform. I will show you one case. Most importantly, because we have that knowledge-based findings, we can understand and predict and generate hypotheses. And I think IPA is strong in that. We can generate hypotheses that obviously you need to test. You always need to validate them in the lab. So we are going to use mechanistic network and regulatory affairs to show you a couple of examples. We can look at any type of process or any type of disease and look at what are the molecules that are involved into that and associated using the bioprofiler. I will not mention that today, but I can do that on a one-on-one. We can compare within particular data set up to 20 observations. More importantly, and I will show that this afternoon, we can compare your own analysis to pre-computed land data set using analysis match feature, which is releasing in the end of September. And that's really the power of the integration with a Microsoft here. We can also incorporate findings and that only you can see with your own login and not being shared with others. So doing that, the data that has been sent when you by Microsoft arrive here. So you have a data set. The analysis can be set up or not, as you wish. But the setup of the analysis, when you open the analysis, is include that type of windows. You have multiple tabs, and all the tabs correspond to what I expressed: canonical pathways, upstream analysis, and I will show you a couple of them: disease and function, regulatory effects, and so on. At the end of September, you will have another tab that I will show this afternoon called analysis match. So this is a summary tab, right? You have the top canonical pathway based on the p-value overlap, couple of upstream regulators, couple of causal networks. All of that can be explored further. So the analysis I'm going to talk about today is at the transcript level, fold change of 2, p-value over 5, and a max of 10 in either the control, which is the responder, or the sample, the non-responder. This is a typical base, like everybody understand, Ingenuity pathway analysis, look at pathways. We look at that by overlap. We can look also by activity. So this is an overlap. In red, all the genes that belong to that particular pathways that are present in your data set and are upregulated, and in green, downregulated. It's a very simple color code. One of them, it's very interesting to see that, in that non-responders, we have the regulation of the EMT. The EMT is basically the step that allows the cells that are attached to each other in epithelial junctions, and they start to lose the E-cadering, as Joe mentions, the expression going down, and allows the cells to detach each other. And that's bad news, because when they are going to detach, they will acquire a new phenotype called mesenchymal. That will allow the cells to be very motile, very invasive, and they are going to colonize distal organs. These steps, you want to stop it if you can. I mentioned also that we can look at overlap, but we can look also of activity. We can predict, based on a couple of very important molecules within one pathway, if a pathway is what we call turn on, which corresponds to a positive z-score, or turn down, which corresponds to a negative z-score. And here, this is a couple of examples: ROA, integrin, PAC, I think cytoskeleton. Everything here is telling me, looking at that there's something about cytoskeleton signaling and EMT. Nothing is surprising. This is the epithelial to mesenchymal transition pathways, part of it at least. And what we do here is we overlay the expression of your gene in your data set. Green downregulated, red upregulated. And you see also the color, blue and orange. This is the prediction based on the pattern of expression of your genes. You can predict that in that case, the mesenchymal phenotype will be increased and the cell-cell adhesion will be decreased. So we are really in the context of an EMT structure, EMT phenotype here. So that's just one aspect of the canonical pathways. We can go a little further. Based on the pattern of expression of genes that are known to be downstream of a particular or associated to a particular phenotype, we can look at that tree map. It's a lot of orange and blue. Everything that you see in orange is predicted to be increased. Everything you see in blue is predicted to be decreased. And you can go down. In every square here correspond to a subset of genes that are associated to that particular pathways. And when you look at that, and you look at what's predicted to be increased, you realize that there's a strong tumor progression signatures. This is not new here in that paper, but we can explore one. Let's look at that progression of tumor. I selected this one because 50 genes are associated to that particular processes, and I can display a network. I could have choose metastasis and everything, but there is more genes, so it will be crowded. Now I put that progression of tumor to the pathways. I try to organize that in a subcellular way, makes a little bit more sense. And as you can tell, based on the pattern of expression of all these genes that are downregulated or upregulated, and you will recognize a lot of them, progression of tumor is predicted to be increased. Okay. Now you can add another functions if you want, statistical functions. And I selected one, and the function that I've seen based on a p-value is necrosis of tumor. And that one, the necrosis of tumor, if you use that tool, the molecular activity predictor, you realize it's predicted to be decreased. So everything makes sense right now. In a non-responder, progression of tumor is increased, necrosis of tumor is decreased. We can explore a little further. Joe was mentioning the e-cadering, CDH1. CDH1 in that particular network is here. So I enhance that view. It's tied to necrosis of tumor by findings, and it's tied to progression of tumors. And here, when you click on that genes, you realize the molecular summary, and you have a lot of splicing variants that are differentially expressed. Not all of them are significant, so I open the isoform view, and we realize that the CDH1-001, according to the ensemble mapping model we have used, is the one which is significantly expressed, that molecule. You can click a little further. If you have findings, we will have an hyperlink here in blue. You click on that, and you will have the finding corresponding. Again, here, nothing is surprising. CDH1 goes down, EMT is up. Let's look at the underlying program. And we use the tool called upstream regulator analysis. And we are going to select, so we are able to, based on the pattern of genes that are known to be downstream of these different regulators. And the regulator can be of different type, can be cytokines, growth factor, kinase, or transcription regulator. And here I'm looking at what has been predicted to be increased or activated, right? And for me, everything tells me this is a complete signature of EMT. To get a proof, I take all the genes that are predicted, the transcriptional regulators are predicted to be activated, and those that are predicted to be inhibited. And you put them on the pathways, right? So this is everything in my data set that has been predicted to be increased, everything in my data set that's been predicted to be decreased as transcriptional regulator. And you add functions, and you look at the functions. 3 of them appears: invasion of cells, angiogenesis, and EMT. All of them are predicted to be increased, and this is the overlap. So right there, even if you didn't know, you can look at what's going on based just on the program. Let's go a little further. We can use, so in IPA, we have different types. We have different network types based on different stringency and relationship between regulator or targets. But let's look at one. This is called the upstream regulator that I just defined, right? The upstream regulator that we mandate that each of these guys are tied to downstream target, to at least 4 downstream target by expression, transcription, or protein bindings. The reason why we do that because this is a stringency that we can infer the downstream effect. We can say if it's active or not. And this is the case. Going back to the upstream regulator I've showed you, I selected 2 transcription factor, twist1 and SNADE2, that are predicted to be increased, it's in orange, and one which is predicted to be decreased, SPDF. Well, this guy is involved in cellular migrations, and when this guy is predicted to be decreased, cellular migration progress. So here, everything is telling me that that program pushed the cells to detach to each other and to migrate. And you will recognize many of them. This is a typical hypothesis. These are the real expression in your data set, and these are predictions. Well, you can test that in the lab and validate. If it's good, that's a good hypothesis to have run. Let's talk about another type of regulator. We have selected transcription regulator. We can look at growth factors. Joe was mentioning FGF2. FGF2, strangely enough, transcriptionally is literally downregulated, but functionally, based on the pattern of all these genes, we believe the program has been activated. And this is one of them. So I put FGF2 in the center and all the genes with their relative expression here. It's really interesting that in IPA you can discover so many things, including the isoform, as we mentioned. In a recent paper, three isoforms with different molecular weight have been involved in melanoma at different steps. And we do have these isoforms here. They are all differentially expressed here, and this is the corresponding. You can click on this finding and go further. Now let's look at a different type of network. We know that one upstream regulator cannot work alone. They all always work together. It's like a Microsoft and IPA. We work together to understand really what's going on. And this is the case. I'm going to use that typical mechanistic network. It's basically mandating that the upstream regulator and the other connected regulators are tied with this type of edges: activation, inhibition, translocation, so on. And all of that network of regulators sits on the target downstream, and they are connected uniquely by expression, transcription, and protein A binding. Therefore, we can infer. And this is one example. If you look in the tab, you have that CXCL12 mechanistic network. And here, this is a network that represents 18 collected regulators. And they are sitting on the top of 511 targets. That means that that network with that predicted activated set of a molecule here plausibly explain the expression pattern of 511 targets. I show you here only 12 growth factors here. Why do we do that? Because that's an hypothesis. And we have a tool called Molecule Activity Predictor, and I can mimic the inhibition of CXCL12 that you see here. By doing that, virtually, this is virtual right now, I can shut down that molecule, I can downregulate or show mimicking the inhibitions. By doing that, you virtually downregulate some of these regulators that are important and also have an impact on the target downstream. Well, this is an hypothesis to test in the lab. That's an hypothesis that is very interesting because you can dissect any of these connections here. I selected CXCL12 because if you click on all these findings, you realize that CXCL12 is the ligand for CXCL4, and that molecule is heavily involved in metastasis and tumor growth. So it's heavily looked at. So in that context of the non-responder, maybe it's a good idea to look at that hypothesis in the lab. Finally, remember, we talk about these upstream regulators, how they are connected to each other. Well, we can now look at a regulator effect which tie upstream regulator and the downstream effect analysis that I showed you at the beginning, the progression of tumor, together, right? They are matching, and they are tied and connected by these downstream targets in your data set, right? So we tie and connect the regulators with the functions via the molecule. And this is one example. Here you have 3 upstream regulators. 2 are predicted to be activated. 1 is predicted to be inhibited. They are all connected to this target downstream here with their upregulation or downregulation. This is CDH1 again. And there is a massive impact on biological processes: invasion, cell movement to tumor cell line, and migrations. So it tells me that that transcriptional program by these 3 guys can drive this process up. Well, these processes are interesting to shut down in cancer, right? And for each of these guys, I have mentioned why they will be important to be counteracted. One way to do that is to use our tool, the Molecule Activity Predictor, and I mimic the inhibition of LOX, mimic the inhibition of CCL5, and mimic the upregulation or the activation of PDCD4. Virtually, you revert a lot of the expression pattern of your gene in your target downstream, as well as the biological processes are reverted. So this is one idea of using IPA in the lab. You generate hypotheses based on real content, real description of the data, and now you can test that in the lab. So I went a little fast, but this afternoon I will be pushing a little further to really show you how we are going to integrate, how we are integrating actually IPA and Omicsoft at a very powerful level. I'm new for Omicsoft. I knew Omicsoft 6 months ago, and I think we have achieved a lot, and this is just the beginning of a very strong connection here. But for what I've showed you today, using RStudio, we can do the RNA-Seq analysis. We can align to the genome of interest and the model of interest. We can perform really strong stat and QC. We can obtain all differential expressions between the samples, and you can send the data, which is really core to me. I'm not a bioinformatician. I'm a biologist. I'm an immunologist of background. The data is sent directly to IPA with the plugin that Joe has mentioned. And in IPA, we can visualize these differentially expressed genes in a non-responder versus responder. We can understand some of the pathways that are involved. They are hypotheses, but they are good hypotheses. We can discover potential transcriptional program, see how these guys are connected to each other to drive the expression of the genes downstream. We can visualize these specific isoform splicing variants that are very interesting for cell movement or tumor progressions. You may imagine them to use them for biomarker application. You can discover some biological processes that participate in that anti-PD-1 innate response. And more importantly, you can highlight new hypotheses. And that is really strong because based on that, you validate, you have your work cut for parts, and you can eventually publish. And that's what I've been doing as an example in using IPA for the last 5 years. So I would like to thank you here.